Ena Naito.
Yeah, thank you. So first of all, thank you to all the speakers so far for the amazing presentations today. And of course, the conference organizers for inviting me to the conference. My name is Ena, and I am a doctoral researcher at Aalto University in Finland. And I'm also a team member of a EU project called CoCoon, where we are developing educational modules to accelerate the green transition. And we're trying to mainstream biodesign education within and across Europe. So my own journey into biodesign started off by being a designer in the lab when I collaborated with 3 other chemical engineers to develop a packaging solution for fresh fruits and vegetables. And we had this amazing opportunity to present our work at the Biodesign Challenge in New York in 2021, where this gained a lot of community and media attention. And one option for us was to continue this as a startup. But what I really, really enjoyed and found value in was this interdisciplinary collaboration and being able to share a language that is biodesign with people outside of my discipline as well. And biodesign also allowed me to approach scientific knowledge like material science or like biology or like chemistry in very designerly ways. And this was very, very fascinating because I wanted to work towards opportunities to create this kind of similar experience for other people as well. So after graduating from my master's, I decided to pursue my doctoral research not anymore in developing new kinds of materials, but more towards creating conditions for other people to be able to get their foot also into biodesign. So now in my research, I'm looking at a bigger picture view of biodesign. And I do this a lot by visiting different labs around Europe who engage with biodesign, to talk with people who engage with biodesign and other fields as well. And specifically, I look at where biodesign happens, whether it's in a science lab, a classroom, a makerspace, or a studio, or even your own kitchen sometimes. And also how biodesign learning happens. So what kinds of educational methods and practices can allow entry-level work into biodesign? And how can we make it accessible to people also who might not have a scientific background? And at the same time, I'm taking all these learnings that I learned from the people I visited, the places I visited, and all the amazing researchers that I've had the privilege to work with to develop and run the bioMakerStudio at Aalto University. And this is a fabrication space where students and researchers and communities can creatively work with biology-themed projects. And these QR codes that I will be presenting throughout the presentation will also be available on the recording, hopefully, later as well. So now I thought for this talk, I would start with the question that's raised in the conference and which has been already quite discussed in other conferences as well, but it's very, very relevant for the biodesign field. So how to grow a biodesigner? But then I thought, okay, this question is very, very complex. It's very hard to unpack. So I just decided to simplify the question to how to grow a plant, which is much, much simpler, right? And then, of course, we are in 2025. The next thing I did was to ask this very question to ChatGPT. And it kindly told me that you need to choose the right plant, you need soil, you need water, you need light, you need fertilizer, and then to maintain and to take care of it. And this is going somewhere because it shows that growing a plant is not just about the seed, but you have to create the right conditions and continually surround with it in order for the plant to actually grow. And I see growing a biodesigner as being very, very similar to growing a plant. So yes, it's about the people, yes, it's about the learners, yes, it's about the students. But whether or how they grow depends on the surrounding conditions that they're provided with. And my own research looks exactly into that. So what are these surrounding conditions that are needed for biodesigners to actually grow? So today we'll tackle these 3 questions: why grow biodesigners, and then where to grow biodesigners, and what next? So how can the biodesign field evolve into new learning opportunities? So jumping into the first question, why grow biodesigners? Why learn or teach biodesign in the first place? And as you might know, there's a lot of discussions in biodesign around its potential for sustainability, for interdisciplinary collaboration, new kinds of prototyping, experimentation. There's also provoking questions on ethics and so on and so forth. But these can be very, very, very broad themes that could be tackled through other fields as well, not just biodesign. So the question is, what makes biodesign actually quite unique? And looking at how institutions and educators approach biodesign learning, we can see biodesign as a tangible way to rethink our current societies. And there are three major rethinking approaches that current biodesign education focuses on. And these are rethinking materials, and rethinking systems, and then rethinking relationships. And I'll briefly explain their approaches in the next few slides. So the first, most common one is using biodesign to rethink unsustainable materials. And this includes, for example, innovating new kinds of materials that replace plastics or other fossil-based materials or animal-based materials. And in this context, many designers are collaborating with scientists to optimize material functionalities and also aesthetics. So for example, biodesigners can experiment with growing mycelium for architecture, like testing fire resistance properties or how to grow bigger and stronger structures that might withstand the weather. So through biodesign, we can learn and teach about participating in designing new and sustainable material possibilities. And then expanding from this smaller material scale to a more bigger system scale, biodesign can help us rethink linear systems. So taking the example of mycelium again, we can think about how can we reuse regional waste streams or byproducts from the industry to grow the organism? Or how might growing as a manufacturing method change how we produce and consume and dispose of things? How can biodesign help us think about the circular economy on a more tangible level? So through biodesign, we can learn and teach about ways to challenge existing systems. And then more recently, there's been a lot of discussion around biodesign, how biodesign can help us rethink our relationship with nature and more than human systems. So here, organisms become co-designers in the design process with their own biological temporalities, with their own biological scales, which are different from the human scales that we live and work with. So for example, unlike working with concrete or unlike working with plastics, many organisms take weeks or even days to grow. And during that time, you have to take care of its well-being in order to co-create this design outcome together with the organism. So through biodesign, we learn to move away from this more anthropocentric thinking to form a more nurturing and caring relationship with more than human lives. And these are by no means the only ways to approach biodesign, and they can also be combined in very, very different ways. But this idea of rethinking through biodesign allows us to understand how different institutions might approach biodesign as opportunities for teaching and learning. But then now this raises the question of how do we practically create conditions for these rethinkings to happen in biodesign? And one big factor that plays a role are the particularities of spaces and infrastructures that allow biodesign learning. So where to grow biodesigners, and what kinds of spaces can we actually set up to learn and teach biodesign? And here are some of the places that I visited as part of my doctoral research, and all these spaces are set up for biodesign, but as you can see, they come in so many different shapes and forms depending on their institution, the users, the lab managers, the funding, available spaces, so on and so forth. But one way to understand the tendencies of these very different learning environments, in my research at least, is by putting them on a spectrum from more controlled spaces to more exploratory spaces here. So on the controlled side of the spectrum here, you might have biodesign spaces that look more like microbiology labs. They usually focus more on the accuracy of tests, reproducibility, and controlled experimentation, like optimizing biomaterial properties. They also prioritize creating sterile environments, so disinfecting your hands and shoes very rigorously before entering the space, with specialized scientific equipment to allow for more precise control of measurements, and then stricter protocols of how certain experiments have to be done. And this also means that the access to the space is very regulated, so they won't let everybody go in and out of the space very freely. And on the other side of the spectrum here are more of the exploratory spaces where they prioritize open-ended inquiry, improvisation of methods, and creative exploration. So they are more open to biodesign projects that can be more messy, typically with more accessible and familiar DIY or kitchen tools. They follow more of a playful approach, typically followed by more of a what-if question. So what if I do this? What would happen if I do this? And these spaces can be more accessible to a wider range of learners because you don't have to have the specific rigorous training to actually get into the lab. But at the same time, the environment is not suitable for systematic experiments or to achieve accurate results because of this open condition. So even if two spaces on the spectrum are both rethinking materials through biodesign, it might look more like this in a more controlled space, as opposed to more like this in an exploratory space. And neither is better than the other. They both have strengths, they both have their own weaknesses, but it also depends on what kinds of conditions you would like to cultivate through biodesign learning. And this includes asking questions like, who are the learners? Are they university students? And if so, from which fields? Or do you work with children, or do you work with local communities? Who are the educators? Do the lab managers or teachers have backgrounds in microbiology? Or do they come from design? Or do they come from material science, for example? What are the uses? Is the space going to be used for courses, hands-on community workshops, material research, or even material production? And then what organisms and practices do you intend to work with, keeping in mind that certain species or certain protocols might need specialized conditions or specialized equipment? And then how much funding do you have, which is a big question, big question. This determines what kind of equipment or spaces or resources or people you can get. And then what other infrastructures are there around you? So are there opportunities for industrial collaboration, for example, or are there specific institutional regulations or rules that you actually have to follow and to keep in mind? And these are some of the conditions or considerations that really shapes what a biodesign space might look like. And to use the example of the bioMakerStudio that I'm running at Aalto University, I would position it somewhere here, so more towards the exploratory spaces side. So we do have scientific lab equipment and tools that we use, for example, an autoclave to sterilize mediums and tools. We have digital incubators to grow things on Petri dishes as well. But at the same time, the bioMakerStudio is a place to explore biodesign in very messy ways, and to also design things out of the Petri dish, so on a bigger, messier scale. And we also hold courses, lectures, we also host master's and PhD research projects, as well as community workshops that are open to the public. So we have a lot of audiences and users working within this space. And this openness allows us to work with different creative fields as well. So for example, growing materials for costume design, or working with analog films, or working with electronics as well in collaboration with a fab lab. So there is no one blueprint to say this is what a biodesign lab or biodesign space should look like. But the decisions that are made in the spaces create certain conditions, which then create certain capacities, which includes some people and then excludes some people, which includes some practices and also excludes some practices. So as the number of these biodesign spaces continues to increase, we have to be aware of what kind of conditions we are actually creating and for whom, and then ultimately for what kinds of biodesign futures we're looking for. And then this brings us to the what next question. So how can the biodesign field evolve into new kinds of learning opportunities? And first of all, what is often overlooked is the role of educators in advancing biodesign. And at the moment, biodesign is such an emerging field that there are very few educators, if at all, who are specifically trained to teach biodesign. And in the EU project CoCoon that I'm also part of, we are developing training manuals and resources for educators in schools. The QR code is on the screen, probably in the recording as well, which we hope would be a starting point for integrating biodesign into education. And then importantly, it's also a responsibility for the educators to think about how to balance openness and safety. So how can we balance freedom of design explorations with the safety of bioexperiments? And then balancing theory and practice, how can we teach sustainability concepts through hands-on approaches? And then to bridge academia and then the wider world. So how can we teach skills that are actually transferable to the real world outside of this academic setting? And then second, what I think is very, very, very crucial at this moment in time is to strengthen and expand the biodesign network. And there are different networks that already exist, but in most cases, biodesign is still quite dispersed and are happening in different hubs around the world. And one of the larger aims of my research is also to be able to connect these places and people and to foster the exchange of ideas and projects as well. So for example, tips and tricks on how to start a biodesign lab or what kinds of educational best practices you have in biodesign. And this also includes sharing my research and learnings in opportunities like this conference that we are in right now. So let's go back to our first question, how to grow a biodesigner. These how questions, as I said in the beginning, are very, very complicated because it is not one straightforward answer. So today I tackled it by asking another 3 questions, which are firstly, important to think about why do we want to grow biodesigners? Because this kind of shapes what kind of rethinking competencies we want future biodesigners to develop. And then another question to ask is where to grow biodesigners, because the design of physical spaces will inevitably shape what kinds of projects, what kind of practices, what kinds of people are included in this discussion and these activities. And then what next? So what concrete agendas could you have in the biodesign community to develop these fields? So this includes training educators and also expanding the biodesign network in my case. And keep in mind that biodesign is still an emerging field, which means that it's still in the beginning of its growth. So how we shape it now at this moment in time will have a big influence on what the field will become in the future. And if you only take away just one thing from my talk today, I would like it to be this. You can start very, very, very small in biodesign. So in January 2024, I started the biodesign table in a corner of the Fab Lab, as you can see here, which basically consisted of one DIY incubator that we made in one day, and we started just growing bacterial cellulose SCOBY culture in there. And then 1 year later, in January 2025, the table has grown into a whole dedicated space for biodesign, including an outdoor community garden as well, with a big community of students and researchers and local people using the space as well. So this is what I mean by growing. So I talked about all these surrounding conditions, but you actually have to start with a seed. And the seed can be a very simple thing. It can be a single table, it can be a single project, it can be also a single person as well. And to develop this seed, I think resources are very, very helpful. So I want to end this talk with a few resources that I share a lot with my students and a lot with my colleagues too. So some biomaterial databases to see what kinds of materials have been explored already, and many have recipes that you could also try out. Some sources of inspiration, including projects and newsletters and podcasts, And then some biodesign learning and teaching resources for educators, but also for independent learners as well. And then finally, if you're interested in the different kinds of biodesign spaces and their strategies, here's my most recently published conference paper for reference to read more about that. But yeah, thank you so much for listening, and I hope to be able to speak with many of you after the conference.
Great. Thanks, Ena. I think we're running a bit behind on schedule, but I'll take 2 questions if there are.
Thank you for the presentation. I just have a basic question, like how can we define biodesigner, and what is the limit between like a designer and a biodesigner?
That's a very good question. I think we can talk for another conference on this, but well, there are different ways that people define themselves as biodesigners. Some people don't define themselves as biodesigners because they feel like the field isn't developed enough, or they get misrecognized on the medical field of biodesign, so that's more established before. But I think it's ultimately up to the designer to see if they feel like they belong in the discipline that I've been talking about, or all the other speakers have been talking about, if they identify with that discipline, and if they identify with that discourse and the community, they are able to identify as biodesigners working within science, arts, textile design, graphic design, whatever discipline you're part of. So for me, I personally identify as a biodesign researcher because I'm not doing biodesign myself. I'm researching the phenomenon of biodesign at this moment. So I think there are different labels that people like to put on themselves to be able to identify with a certain community, if that makes sense.
Thank you for your presentation. I have a question regarding your teaching resources. I managed to quickly skim through. As you said that there are not so many trainers in the field, how do you make sure, how do you promote these teaching resources if you do? Or it's more like people find you or you find people, like how this is happening?
Yeah, thanks for the question. I think it goes both ways. So yes, I go around and promote the EU project that I'm part of. We are also a combination of institutions. So they're all different hubs that can expand around their network as well. But at the same time, I think people can also proactively try to look for these resources. And I think that comes from more educational institutions being involved in biodesign, whether they make a master's course out of it or whether they make a one-week workshop out of it. And that requires the educators to then start looking for resources. And we want to make that easier for other people to find as well through these projects.
All right. Thanks so much, Ena.